Hello everyone. The topic that I'll be presenting is from third unit. The contents are introduction of antigen-antibody reaction, agglutination, precipitation, immunoelectrophoresis, immunofluorescence, ELISA and RIA. Antigen-antibody reaction. These are a specific, reversible, non-covalent biochemical reactions between antigen and antibody. If we see this antigen and antibody, these antibodies are a smaller and always soluble antibodies, whereas antigen it can be a smaller or soluble or insoluble or a larger antigen. If the soluble antigen combines with the soluble antibody, this reaction is called a precipitation reaction. Whereas the insoluble antigen reacts with soluble antibody, this reaction is called as agglutination reaction. Let's see the various different types of in vitro antigen antibody reactions. They are agglutination, precipitation, immunoelectrophoresis, immunofluorescence, ELISA, and RIA. In agglutination, we have particulate antigens. These are the particles, such as cells, that can carry antigenic molecules on their surface. For example, bacterial cells, fungal cells, into the mammalian cells. For this, when a specific antibody to the particulate antigens are added, this cross-linking will occur, resulting in visible clumpings or agglutination of particulate antigens will happen. The difference between the agglutination and precipitation reactions is that precipitation involves in the aggregation of soluble antigens, whereas the agglutination involves in the clumping of insoluble or particulate antigens. Like precipitation reactions, agglutination also occurs when the antigens and the antibodies are in equal proportions. Agglutination reaction is defined an antigen-antibody reaction in which antibodies cross-link particulate antigens, resulting in visible clumping of the particles. In diagnosis immunology, agglutination reactions have wide variety of applications. It uses for detecting both antigen and antibody in the serum and also in other body fluids. Most importantly, agglutination reactions have been performed routinely in the blood banks for detection of ABO and Rh blood typing in preparation for transfusion. Here we have two types of agglutination reaction. One is active agglutination and another one is passive agglutination. This classification is based on the antigen particulate or epitope to the antibody which it will bind and the present naturally on the target cells or not. Active agglutination. The epitope of interest are naturally found on the test particle. And we know that antigens are commonly found on RBC cells and antigenic determinants are also found in bacterial cells and fungal cells, etc. And these antibodies can directly bind to these antigens and agglutinate them. Let us say that we have the pure bacterial suspensions or culture. From that we are going to make a suspension and we are keeping this bacterial suspension on a glass slide, and for this we are going to add a antiserum which contains a specific antibody for this antigen, and after some time we can see that the clumping as well as clearing of suspensions, that means the agglutination has formed and this test is positive. And this type of bacterial agglutination tests have been performed routinely in the labs because by doing this, the bacteria which have isolated from the clinical specimen can be identified. If you look into the application part, this active agglutination have been used for blood grouping and cross-matching before blood transfusion, and this can be used in diagnosis immunology as well as in the vital test for diagnosis of typhoid fever, and Brucella agglutination for brucellosis, and biophilics test for rickettsiosis. Let's see the passive agglutination. In passive agglutination, the epitope of interest doesn't occur naturally on the cells or particles to be agglutinated. In such cases, epitopes are soluble antigens or chemically fixed to the carrier particles. Few examples of carrier particles in passive agglutinations are latex, polystyrene, and bentonite. And this passive agglutination can be done when the pathogen culture is not feasible and only soluble antigens are present or available. And one of the applications of this passive agglutination is used for diagnosis of bacterial diseases or viral diseases. One of the most important example is latex agglutination test for rubella antibody testing. Let's say that we have a carrier particles. In the first row, we are going to add a antigen of interest, such as soluble antigens. And resulting in we can see that antigens, that is carrier particles, are coated with soluble antigens. In the second row, here we are going to add a specific antibody for these antigens, and resulting in we can see the agglutination reaction by the antibodies are reacted with antigens, and these particles are agglutinated with one another. In the third row, we can see one more method, that is here we are not adding antigen of interest. Instead of, we are going to add antibodies. In this method, we can see the detection of antigens, and hence this method is called as reverse passive agglutination. To understand what is precipitation reaction, let's understand the topic immune complex formation. If we look at this first beaker, here we have an unknown amount of antigen, and we added a known amount of antibody. What will happen in the second beaker is this antibody and antigens or corresponds to each other. Once they come in closer proximally, they will form a non-covalent bond with each other, resulting in the formation of small amount of antigen antibody complexes. And we know that each antibody has two antigen binding sites, hence we call it as bivalent. And we also know that each antigens have epitopes in moreover, and all these epitopes are identical, hence we call it as antigens or multivalent. If we look at this last beaker, we can see the different antibodies are binding with the antigens, with the same antigens, that will make a cross-linking complexes. This repeated type of antibody antigen cross-linking formation or complexes will form a immune complex or lattice. This antibody and antigen cross-linking lattice formation will form a large amount of precipitations which are visible out of the solution. As the size of antigen-antibody lattice increases, it loses its solubility and precipitations out of the solution and thus becomes visible. Now we understand the precipitation reaction. When the antibodies mixed with soluble antigen in equal proportions results in lattice or immune complex formations, which is insoluble, and these insoluble complexes are called as precipitation and they are visible. Let's have a look at the conditions necessary for precipitation reactions. Here we have antibodies which must have at least two antigen binding sites or bivalent, And second condition is antigen must be soluble and it must be either bivalent or multivalent, and at least two epitopes of antigens must be identical. The proportion of the antigens and antibodies must be equal, that which will form a lattice complex. And this reaction occurs in the presence of electrolyte with a suitable temperature and pH. And precipitations can take place in a liquid media or in gel. Let's see how relative proportion of antigen antibody influence the formation of precipitate. Let us say that we have three test samples of soluble antigen in increasing amount. Suppose the first test sample has 2 antigen, second has 6, and third has 18 antigens. For each of the test samples, we are going to add a same amount of antibodies. Suppose we'll see that we are going to add the 7 antibodies in each of the test sample. If in case of first test sample, antibodies are in excess because antigen will bind only 1 or 2 antibodies, hence there is no cross linkages are formed. The presence of the antibodies and the antigens are insufficient, so there is no lattice formation will take place. If in case of second test sample, antigen and antibodies are in optimum concentration because the antigen epitopes and the antibody binding sites are in equal proportion. Each antigens are shared between two antibodies, so there will be a formation of cross linkages, and that will result in the lattice formation, which will form a visible precipitation in second test sample. If in case of third test sample, antigens are in excess, there is no cross linkages because the antigen and antibody complexes are formed, and it is formed in a smaller amount, and there is no formation of the precipitation because it is insoluble. Now we understand the relative concentration of antigen and antibody reactions for the precipitation reaction. Now let us understand the precipitation curve. Precipitation curve, it is a graphical representation of precipitation reaction. In this reaction, concentration of one reactant is kept constant and the another one concentration of second reactant is increased serially. Let us say that we have the series of test sample in the test tube that contain a constant amount of antibodies. In the next step is we are going to increase the amount of antigens. After sometimes precipitate occurs, and after that the next step is centrifugation of each of the sample have done, and then we are going to discard the supernatant and measuring the precipitation. Results can be shown by graphical representation. If you look at this graph, you can see that the x-axis is represented by the increasing concentration of antigen, and the y-axis is represented by the amount of precipitation formed, which contains the antigen and antibody complexes, and this will form a precipitation curve. This precipitation curve will show the increasing amount of precipitation with increasing amount of antigen up to the certain peak. After that, the curve will decrease due to the decreasing amount of the precipitation and increasing amount of the antigen. For further understanding, this graph can be divided into three zones. First one is zone of antibody excess, second one is zone of equivalent, and third one is zone of antigen excess. In the first one, zone of antibody excess, there is no aggregation of antigens, hence there is no lattice formation will take place. In the zone of equivalence, we can see that the antigens and antibodies are formed lattice formations, hence there is a maximum of precipitation concentration will increase. And if you see the zone of antigen excess, again there is no aggregation of antigens, hence there is no lattice formations, hence we can't see the precipitations in this zone of antigen excess. Immunoelectrophoresis is defined as a technique in which an antigen mixture is first separated into its component parts by electrophoresis and then tested by double immunodiffusion. Before getting to the details of immunoelectrophoresis, let's understand the electrophoresis. Electrophoresis is referred to a movement of macromolecule in a electric field. It is a basic setup of electrophoresis. Here we can see that we have the electrophoresis tank and we have gel tray where we are going to add an antigen and antibody reagents, and we can add the buffer solution here, and we can see the two electrodes are connected to the power supply, and the yellow color is shown, that is negative electrodes, and red color is positive electrode. When the electric current is applied, the macromolecules will start migrating. And if you look at the second picture, we can see that macromolecules will migrate, and it is on depends on the on their own charges and size. If you look at the charges, and net positive charges will start migrating towards the negative electrodes, and the net negative charges will start migrating towards the positive electrodes. If you come to the size, then larger molecules will start migrating slowly, and it covers very short distance but in the given period of time. And if you look at the smaller molecules, it will migrate very fastly and it will cover a large distance. And we all know that antigens are made up of the proteins, and they have their own charges and their own size. So they can also migrate in different speed and and it can also cover a different distance also. Let's see the steps involved in immunoelectrophoresis. In the first steps, preparation of the gel-coated slides or glass slab. And for this, we are going to use a supported media such as agarose, because we know that it doesn't have any charges on itself, and it cannot interact between the gel and reactants. Once the gel is prepared, it's poured onto the glass slab and allow it for solidify. Once the gel is set, we are going to cut out a well on the one side of the gel, and again we are going to cut out a toes or the others end of the gel. Once it is done, next we are going to add a mixture of antigen solution on the well. Once it is added, that the antigen solutions will start migrating towards their respective electrodes. For example, the negative charges of antigens will start migrating towards the positive electrodes, and the positive charges of the antigens will start migrating towards the negative electrodes. Once it is done, the gel is removed from the electrophoresis, and then we are going to add an antibody solutions on the straws, and we are going to leave it for a certain period of times, because antigen and antibody will start migrating towards each other, and that will form a diffusions. And after sometimes we can see the curved precipitation bunch at the zone of equivalence. First, we are going to use this gel, and it can be visualized and identified by using the double immunodiffusion assay. Application of this immunoelectrophoresis is it can be used in clinical labs for the detection of presence or absence of the proteins in the serum. Here we have immunofluorescence, and we all know that in this antibodies can be labeled with the molecules that have a property of fluorescence. And this fluorescence absorb a light of one wavelength, and it can emit a light of higher wavelength. And these emitted lights can be viewed with fluorescent microscopes and the UV lights. And we have different types of fluorochromes, such as a fluorescent. It's most widely used in immunofluorescence. That will give a yellow or green fluorescence, and rhodamine that will give a deep red color, and phycoerythrin that will give a red color. And we have two different types of immunofluorescence, that is direct immunofluorescence and indirect immunofluorescence. You can see this direct immunofluorescence. It is a very simple and common procedure. In this, we are going to fix a antigen on the slides, and after that, we are going to layer a antigen with this fluorescent label antibodies and then we are going to wash the slides to remove the unattached antibodies. After this, we are going to observe this slide under the UV light in a fluorescent microscope. If you look at this picture, you can notice that antigen and antibody complex. Along with this antibody, you can see this fluorochrome, and that will indicate that will indicate the specific antigen, and that will appear as a yellow and green fluorescence under the microscope. In this indirect immunofluorescence, we have the unlabeled antibodies that we are going to apply on a tissue section. And when the unlabeled antibodies are present, that will bind with the specific antigens. After that, we are going to add a fluorochrome conjugated anti-immunoglobulin antibody. Because this fluorescent labeled anti-immunoglobulin antibody is highly specific for unlabeled antibodies, hence it binds with the already linked unlabeled antibodies. And one of the advantages of this application is there is no loss of primary antibodies, and this technique is more sensitive. And if you look at this structure, you can see that antigen antibody complexes. In this, the secondary antibodies are attached with the primary antibody, and that has formed a complex with antigen. And these secondary antibodies has a fluorochromes on the top, and that will help to appear in a yellow and green color under the fluorescent microscope. And if you look at the application of this indirect immunofluorescence, you can see that it has been used for location identification of the antigens and the location of hormones, and it also used for the identification of subpopulation of that is lymphocytes, mainly in a T helper cells and T cytotoxin cells. And it's also used for the identification of the bacterial species, and it's also used for antigen antibody complexes in autoimmune diseases. ELISA, it stands for enzyme linked immunosorbent assay. ELISA is labeled immunoassay where the antigen or antibody is labeled with a reporter enzyme. In this reporter enzyme, that will produce a color change when the enzyme reacts with a specific substrate, and that will change the color. And this technique is used for the identification and quantification analysis of the substance. And this is also used for the detection of peptide bonds and proteins and the antibody and hormones in the samples. Here we have the three different types of ELISA, that is direct ELISA, indirect ELISA, sandwich ELISA. If you look at this picture, you can notice the basic difference between the direct ELISA and indirect ELISA. In this direct ELISA, you can notice that enzyme-linked antibody is directly attached with the antigen, and whereas in indirect ELISA, this complex which has a enzyme-linked antibody, it's attached with the antibody, which is not directly attached with the antigen. So this is a difference between the direct ELISA and the indirect ELISA. Direct ELISA. This is a technique. It's used for detection of antigen in a serum sample, and the test results can be positive or negative. It completely depends on this presence of a specific antigen in a serum sample. Let us say that we have taken a serum sample from the two different patients, and we are going to use this or apply this serum sample in a two different microtiter wells. And let us say that we have taken a two microtiter wells, and we have added a antigen or serum sample into them. If you look at this first picture, you can notice that there are antigens are present because these specific antigens are observed and immobilized in the microtitules. And if you look at the second microtitules, you can't see any antigens because there is no observed antigens are present. Anyways, we are going to wash this wells to remove any unbound antigens are present. And the next step is, and we are going to add a enzyme conjugated antibody for this specific antigens. I have already told that we are going to add that enzyme conjugated antibodies into the each of the wells. And then if you look at the second picture, you can notice that these enzyme conjugated antibodies are binding with the specific antigens, and they are making a complex. And again here we are going to rinse the wells to remove any unbound antibodies. Hence, if you look at the last picture, you can notice the complex formed by the enzyme conjugated antibodies with the specific antigens. And if you look at the second picture, like a second well, there's no complex has formed. Hence, there is no specific antigen has been present in the serum sample. And the last one, that is the third step, that is substrate. Here we are going to add a substrate because the enzyme that will react with the substrate and that will form a color complex. So we are going to add a substrate in each of the wells, and you can see that in the last picture there is a color formation has done because it indicates that there is a specific that specific antigen has bind with the enzyme conjugated antibody, and that enzyme has reacted with the substrate, so hence it has changed the color, and this test and this color has indicated that the positive test. And if you look at the second one, there is no color formation has formed because there is no enzymes has reacted with the substrate, hence it is a negative test. And mainly, this test is used for immunohistochemical staining of tissue and cells. Indirect ELISA, it's the most popular technique and it's mainly used for detection of specific antibody present in the serum sample. For example, for antibodies to HIV, rubella virus, and it's also mainly used for detection of certain drugs present in the serum sample. And this technique is mainly used for detection of the specific antibody present in the serum sample. Let us say that we are going to take two serum sample from the two different patients, and we are going to detect a specific antibody present in the serum sample. So we are going to take two microtitules, and in that we have added a specific antigen to the antibody of its interest, and that will absorb and immobilize it in the microtitule. And you can see that the antigens are already present in the wells because for example, we are going to detect the antibody for the HIV virus. So in this, the antigens are already absorbed in the microtitules and have formed a recombinant envelopes, and that is a core protein for the HIV. And in this microtitules, you can see the unbound antigens are also present. So we are going to rinse this wells to remove the unbound antigens present in the microtitules. And if you look at the second step, here we are going to add a serum samples for the each of the wells. So you can see that the antibody of its interest, that is antigens, are added into each of the wells. And if you look at the second picture, you can notice that the antibody have already bound with the antigens. And you can also see that unbound antibodies are also present in the microtitules. And if you look at the second picture, that in that the second well, there is no antibody has found bound with the antigens. Hence, that is not a specific antigen for that antibody. And we also call this antibody or is that is primary antibodies. And again, we are going to rinse these wells to remove unbound antibodies which are present in the wells. And if you look at the last picture, you can see that the primary antibody has made a complex with the specific antigens. And if you look at the second well, there is no complex formation has done. And in the third step, we are going to add a enzyme linkage, and that is secondary antibody. We call it a secondary antibody because that has an antigen binding site for the for the antigens that has a Fc region for the primary antibody. And then you can see that this enzyme linked secondary antibody that has attached to the primary antibody, which has already attached with the antigen, that is a huge complex has formed in the secondary that is second picture. In that first well, you can also see that unbound secondary antibodies are also present in each of the wells. And if you look at the second picture, that is the second well, you can see that the unbound secondary antibodies are present that has not attached with the antigen, because there is no primary antibodies are present in that second well. So again we are going to rinse this well to remove the unbound secondary antibodies. So after rinsing it, you can see that in the last picture that this first well has a complex structure which has a secondary antibody. Primary antibody has already attached with the antigen, that is specific antigen. And if you look at the second well, there is no complex formation, so it is a empty well. And if you look at the last step, that is substrate, we know that the enzyme will react with the substrate to form a color complex. Here, that enzyme is secondary antibody. So we are going to add a substrate in each of the well. And if you look at the last picture, you can notice that a color formation has done because of the secondary antibody has reacted with a substrate, and it has formed a color change. So this test is declared as a positive test. And if you look at the second well, there is no color formation has done because there is a absence of primary antibody. Because of that, there is no bound or attachment with a secondary antibody. So there's no enzyme is present, so it can't react with the substrate, so there's no color change has formed, so it can be considered as negative test. And earlier we have already discussed the direct ELISA and indirect ELISA. Now we got to know the difference between these two. Here we can see that the conjugated primary antibody has attached with the antigen, that is a small complex, and that we have added substrate, and again the color formation has done. And if you look at the indirect ELISA, here we have the primary antibody between the secondary antibody that has a enzyme conjugated antibody and the antigens. And again here also we have added the substrate, and there also it has a color change has happened. So this is a difference between the direct ELISA and the indirect ELISA. Sandwich ELISA. It's mainly used for detection of antigen, and one of the most important application is for home test kits. For example, home pregnancy test kits. In this sandwich ELISA, we are going to use two different types of antibody for the same antigen. In this, antibodies will bind to the different epitope present on the same target antigens. For this, we need three components, that is capture antibody, target antigen, enzyme linked antibody. Sandwich ELISA is an antigen is trapped between the two different antibodies. The first step is we are going to add a capture antibodies into the microtubule well. If you look at the picture, you can see that a capture antibody is already observed and immobilized in a microtubule well, and you can also notice that a unbound capture antibody. This capture antibody is a high affinity for the target antigens. So we are going to rinse this well to remove the unbound capture antibodies. And second step is we are going to add a serum sample which contains a specific antigen. If you look at this picture, you can notice that many different types of antigen is also present with the different types of epitopes. And if you look at the second picture, you can notice that that is capture antibody, it's binding with different types of epitopes of the same antigen, and along with that you can also see that unbound or non-specific antigen also. So we are going to rinse this well to remove this non-specific antigens, and you can see that only antigen and capture antibody is only present in the last well. And step 3 is enzyme linked antibody are added, because if you look at the picture, you can see that the enzyme linked antibody is also going to bind with the antigen, and along with that you can see that unbound antibody is also present. So we are going to rinse this well to remove this unbound enzyme linked antibody, and you can see that in the last step you can notice the complex formation has done. And at the last step we are going to add a substrate where the enzyme will react with the substrate and will form a color complex. And here we have added a substrate, and if you look at the last well, you can notice the color formation because that the capture antibody has bound with the antigen, that is a target antigen. Again, the target antigen has already bound with the that is enzyme linked antibody, and this enzyme has reacted with the substrate, hence there is a color formation, and this test is positive. Radioimmunoassay. It's an in vitro assay technique for detecting an antigen or antibody present in the sample. Here we are using the radioactive substances. The principle behind this via is competitive binding. In this method, an unlabeled antigen competes with a radio-labeled antigen for an antibody. This determines the concentration of the unlabeled antigen. Here we have the three different types of principles for the, it depends on the immune reaction, for example, antigen antibody binding, a competitive binding, and the measurement of the radio emission. And for this, we are going to use a radioactive iodine or a tritium can be used for the labeling of antigen. In this procedure, if you see that we are going to add a known amount of antibody on a solid phase. For this, we are going to add a same amount of radioactive labeled antigen, and again we are going to add a test samples which contains a unknown antigens, and we are going to mix this whole substances, and after that we can see that the labeled antigen and unlabeled antigens will compete for the antibody binding sites. If greater the test antigens will bind to the antibody, the lesser the label antibody antigen will bind to the same antibody. This antigen and antibody complexes will get separated by the double antibody techniques because it combines with the polyethylene. Along with that, we also use this charcoal, cellulose, chromatography, or a solid phase technique for the separation of antibody and antigen and antibody complexes, because that will help to form a standard curve which has a increasing concentration of unlabeled antigen to fix a quantity of labeled antigen and antibody complexes, and the presence and the amount of antigen in an unknown sample can be calculated from this curve. This is an flowchart of radioimmunoassay procedure. As I told you earlier, we are going to add a known labeled antigen along with the patient sample. In this, we are going to add a antibody also, and after this, we are going to incubate this for some period of time, and later this known antigen and the unknown antigens will try to compete for the binding site of the antibody, and later the antigens that is unknown antigen will try to bind with the antibody, and the label antibody will bind very in a lesser amount, and this can be calculated, and the standard curve has been already formed, and that will give the unknown concentration of the antigens present in the serum sample. You can see that in the in this curve which we have mentioned here. By this curve we can calculate the concentration of the unknown antigen. Let's see the disadvantages of this radioimmunoassay. That is, this can cause a radiation hazard, and it requires a specially trained persons. And this labs requires a special license to handle this radioactive materials. And it's also required a special arrangement such as like handling, storage of radioactive materials and radioactive waste disposals, etc, and it has a short half lifetime of the isotopes, and it's very expensive instrumentation for the counting of radioactivity. If you look at the advantages of that is radioimmunoassay, it's very specific and sensitive technique, and the application part, it's mainly used for analysis of hormones, vitamins, metabolites, and diagnostic markers, and it's mainly used for detection of early stages of cancers, and it's also used for measuring the concentration of toxins present in the contaminated foods, and it's also used for detecting the infection caused by the sexual transmitted agents like HIV, chlamydia, or hepatitis B and C, and it's also used for detecting the drug poisoning such as like cocaine and other drugs. Thank you.